Hello, I'm Paola Dama, a founder and CEO of NAMina Bio. NAMina Bio is building an integrated ecosystem that connects advanced human relevant models, dynamic experimental systems, engineering, and data intelligence to generate more reproducible, interpretable, and decision ready translational evidence. In organ-on-a-chip and complex 3D workflows, biological meaningful signals can sometimes be difficult to distinguish from technical variability, particularly when the expected treatment effect is relatively small. Through our collaboration with the UstarFlowAI, we are exploring how AI-enabled assay-confidence modeling and standardization can help address this challenge. We here want to show where UstarFlowAI fits within the broader NAMina Bio workflow. We begin with the human relevant model, such as an organ-on-a-chip systems, tumoroids, or another dynamic 3D platform. These systems generate complex assay data that may be influenced by biological response, flow conditions, batch effect, and well-to-well variability. UstarFlow AI is being developed as a confidence layer that can help characterize variability, support assay standardization, and assess whether a biological signal remains reliably detectable. The goal is not to replace biological interpretation, but to strengthen and support better compound prioritization and more decision-ready evidence. UstarFlow AI is currently under research and development, with its initial framework evaluated through in silico modeling and experimental validation, represented the next important stage. I'm now pleased to introduce Shrawasti, who will explain the scientific rationale, the proposed approach, the simulation framework, and the next steps for validation.
Thank you, Paula, for the introduction. I'm introducing UstarFlow AI, where we are fixing assay noise in organ-on-chip drug testing. The odds are really brutal. Every year, pharma companies accidentally throw away potentially billion-dollar drug leads because preclinical assays such as organ-on-chips is too noisy to detect the weak yet biologically significant signals. As regulatory pressure mounts to move towards human relevant science and these chips are scaled, the manufacturing flaws further increases the risk of false negatives and also the risk of throwing away viable drug leads. Promising drugs may be deprioritized simply because noise made them look like a failed compound. UstarFlow AI is an infrastructure software and a standardization layer which sits on top of existing barrier organ-on-chip workflow at a crucial lead optimization stage and to decide whether to advance or deprioritize compounds. By standardizing physical variables of the chip, assay's technical noise is reduced and signal to noise ratio is improved. As a result, assay's detection floor is lowered to make small biological signals consistently and confidently visible, to reduce false negatives and rescue drug leads. We performed an in silico analytical validation using 10,000 iterations of Monte Carlo simulation. Our engine optimized the internal blueprint of the barrier organ-on-chip device and minimized the propagated technical noise due to manufacturing defects by up to 47%. As a result, confidence is installed in the preclinical data and the risk of false negatives is reduced. Now let's look at how we frame this study using a formal regulatory mindset. To establish the credibility of our computational model, we defined a clear context of use and set rigorous boundaries to see standardization actually solves the industry's noise problem. The core objective here is the defined context of use as noise completely swallows subtle but biologically significant drug effects. We wanted to see if our standardization could reliably rescue these drug leads by cleanly resolving a tight 10% permeability shift. We stress tested the math against real-world chaos across a 10,000 iterations of Monte Carlo simulation. First, we assume standard 3 sigma injection molding manufacturing tolerances for the chip itself. We fixed our throughput at 48 replicates. Then we threw massive biological variations at it, sweeping run-to-run biological fluctuations across a wide 3 sigma envelope of plus minus 20% to 30%. Crucially, we also dynamically shifted the split of systemic noise to independent well-to-well noise from 25-75 split all the way to 40-60 split to account for different environmental and pipetting variations. Our target operating point sits at a nominal total assay noise of 17.5% CV. Our decision rule was strict and binary. For detectability to pass, the 10% permeability shift had to sit completely outside the uncertainty ribbons across every single one of those chaotic parameter combinations. If the ribbon overlapped the 10% shift even once, it was a failure. In practice, they realized as a noise for a given run or a day differed from its nominal noise. Ribbons represent that uncertainty in the noise. As seen, blue ribbon for standardized barrier OOC is structurally spooled downwards compared to gray ribbon. This puts 10% shift comfortably outside the blue ribbon versus inside the gray ribbon for the current industrial standard barrier OOC at a nominal assay noise of 17.5% CV. We successfully validated that the standardized barrier OOC assays possesses the sensitivity required to reliably and consistently resolve a 10% permeability shift from its background noise, a threshold that remains completely obscured by the uncertainty ribbons of the current industrial standard alternatives. The future of drug discovery depends on the human relevant data we can trust, and UstarFlow AI is the essential bioinfrastructure making it possible in the FDA modernization era. Thank you for your time.